Hi, everyone. Welcome to our second session of GRI University. So today we are going to be talking about neuroscience, GRI receptors, and personalized research. And let's see, we wanted to thank our sponsor, Homology Medicines, for sponsoring GRI University.
And actually, if you go back for a second, Megan, I also just wanted to say a little bit more. So we're super grateful for Homology Medicines to sponsor GRI University, but they've also been a great friend to GRI research in other ways. So I met with them first 2 years ago. I guess it was 3 years ago in Boston, and talked to them about my son and grin disorder, and asked if they would let us try. So this is a gene therapy company, asked if they would let us try their medicines on GRI mice. And at the time they agreed. And so we've connected them with researchers, and it took a while to get it all off the ground, but we've now started, Dr. Amy Ramsey at the University of Toronto and Dr. Graham Collingridge at the University of Toronto have just recently started trials on GRI mice using the gene therapy provided by Homology Medicine. So we're grateful for them for that, and we're grateful for them to be the sponsor here today.
Wonderful. All right, so just to go over the goals for this session. So essentially, we understand that our families come from all kinds of different backgrounds. Everybody has a different education level and different experiences. And last time I thought it was really valuable, Keith asked everyone at the same time for us to input. Oh, looks like we have somebody, if we can mute. Some clinging in the background. But yes, so last time we put in the chat everybody's education level, which was super helpful to kind of gauge the audience and see where people last dropped off with their last science classes. We had lots of people who said that their last science classes were in high school, some were in college. So we understand that everybody comes from a different starting point. So if you want to put in the chat now, what was your last science course? Okay, so 9th grade, A&P in college. High school biology and geology. Perfect. So again, we are working to help families prepare for the complex discussions that are going to take place at our virtual conference and beyond. So this is supposed to be a safe space for everybody to learn and grow together and understand more about GRI disorders. So just to recap from last time, so that session worked on the basics of genetics and how it relates to GRI disorders. So we had Dr. Lemke and Ariske Liston, who spoke to us about prevalence, symptoms, treatment, a little bit about genes and proteins and genetics. And so if you weren't there for the first session, you can feel free to go over to Kiergren's YouTube channel to check that out. And today, what we are going to be focusing on is loss of function, gain of function, and why that matters. So we're going to start with an intro to neuroscience and how the brain works, and then go into GRI disorders and the receptors that are involved. So we want to thank Dr. Coons and Dr. Swanson for joining us today to lead this discussion. And so a little bit of an overview of how this is going to flow. I'm going to give a short talk. Then Dr. Coons is going to talk to us about GRI receptors. Then Dr. Swanson is going to talk to us a little bit about personalized research and medicine, and then we'll have a closing for the session. So as an intro to neuroscience, our brains make up about 2% of our body mass. So when we think about it, something that weighs, about 3 pounds in the average adult, it makes up about 2% of our body mass, but uses about 20% of our body's energy. So they are very metabolically intensive little organs, but they're so important for how we function. And there are different parts of our brain that control different things. So I don't know, we'll see if you guys can see here. I'll try to use the mouse. 1 second. Perhaps not. Um, so the first section that you can see in red on our little brain diagram is our frontal lobe. So this is an important region for cognitive functioning. It is very important for higher executive function, like the regulation of emotions, how we plan, reasoning, and problem solving. All of those things take place within our frontal lobe. Then we have our parietal lobe, which is going to be important for information about temperature, taste, touch, and movement. And areas in the parietal lobe are also important for processing of that information. So that, for example, the parietal lobe can discern between two different objects. And like if you were to touch the skin at different points, it can tell distinctly whether that is at one object or separate objects. And then we also have the temporal lobe down here. So the temporal lobe is near our ears, and it's really important for the sensations of processing sound. It is also important for hearing, recognizing language, formation of memories, and it also integrates a bunch of our senses as well. And then at the back of our brain, we have a region called the occipital lobe. And this is our visual cortex region. So there's a cortex in there called B1 that receives visual information from our eyes. And that information is relayed to different visual processing centers, which can interpret things like depth, distance, location, and be able to identify objects that we see with our eyes. So a little bit about neurons. So we have 86 billion neurons in our brain. And if we were to line all of them up, it would take about 1,000 kilometers, which would be about 200 hours or 8.3 days to walk across all of our neurons. And we actually have more neurons in the brain than there are stars in the Milky Way. So they are very important cells, and they are the messenger cells. So they work to send and receive and process information in our brain. They are the wiring system of our brain, and they communicate by sending signals to each other to allow us to do activities like walking, seeing, thinking, hearing, all of those great things. And here, we have a cartoon of a neuron. I also have a fun little stuffed animal neuron here that we can kind of talk about the different parts. So what makes a neuron unique is that it has these long arms. So these are our dendrites. They are going to receive information from an incoming neuron. So you can see down here that we have the dendrites up at the top. So that's going to receive information. Then we also have the cell body. So the cell body is going to be like the middle part where we also have our nucleus. And that is going to be important for processing information and integrating information. And then we have our axon, which is this long piece down here that's going to send information down. So information is going to travel down the axon. And then at the bottom, we have these little axon terminals. So the axon terminals are basically where information is going to be received down at the bottom. And then if we have neurotransmitter, it's going to be released into the synapse. So we'll talk a little bit more about the synapse. But basically what neurons do is they're sending signals from our brain and our spinal cord, from the brain back to the body, from the body to the brain. And these long axons, the longest one in the body actually extends from the bottom of the spine down to your big toe. And in adults, that's about 3 feet long. So we have some really long axons in our body, and we also have some very short ones. So it depends on where the information is traveling to in the body. And neurons send information to each other electrically, like a computer circuit, or by releasing chemicals that activate other cells. So those chemicals would be things like neurotransmitters. So basically what happens is a neuron gets stimulated. It's going to generate what we call an electrical potential, and that's going to travel down the axon. When that reaches the end of the cell at the axon terminal, that's going to prompt the release of neurotransmitter down there. So that's going to go into the synapse, which is a small space between the two neurons. And it's just a gap that's going to allow the signal to pass from one neuron to the next. So the neuron must become activated, which happens when a neurotransmitter, which is basically like a chemical messenger, that's going to bind to what we call a receptor. So for example, when glutamate and glycine, which are two types of neurotransmitters, bind to the NMDA receptor, it's going to activate the receptor. So when a receptor is activated, that means that it's going to basically open. So we can see that in our cartoon here. So we have this one purple neuron on top, and we have these little tiny blue dots in these vesicles that's going to be released into this space. That space is that synapse that we were talking about. And then we have some receptors down here at the bottom that are opening and receiving that neurotransmitter. So neurotransmitters, there are many, many types, but one of the most important types is glutamate, and glutamate is what we call an excitatory neurotransmitter, which means that it's just going to promote more and more action potentials as that signal gets passed. And we have about 80 trillion of those little gaps or synapses in the brain, and about 90% of those are glutamatergic. So that tells you how important glutamate is, how important the NMDA receptors are, and how important these little spaces called synapses are. So without further ado, I will stop sharing and allow Ian to tell us more about receptors.
Thanks very much, Megan. Am I coming through loud and clear?
Yep.
Yeah.
Good, pleased to hear it. Okay, let's see if I can get this. Let's see if I can get this working. Ooh. Okay, so yeah, so just to add more, I guess Megan's already kind of covered quite a bit of the basics there, and I'll kind of go over some of those bits as well, and as well as some more, perhaps some more slightly specialized bits. So in terms of the ways and the scales with which we can look at glutamate receptors, we can look at them in terms of the entire brain, or look right down in terms of them as individual atoms and individual proteins. So in terms of what you found out about in lesson 1, there was Dr. Lemke, who was talking about this kind of end, where he was talking about showing brain scans, talking about effects on the whole end, the whole brain. And of course, somewhere off the end here was talking about the actual patients themselves, and everything that the defects in glutamate receptors can potentially be manifested in the patients. And then at the other end, somewhere over here, Dr. Liston was talking about DNA and how DNA can be turned into protein, and how the defects in DNA can lead to defects in these proteins. And I guess my job today is to try and kind of bridge the gap between those two. So talking about how DNA forms the proteins the exact way they are, how they come together at synapses, and how they produce electrical signals. And so I guess the main aims are just to a little bit of a recap from stuff in lesson 1, just to make sure you kind of know where I'm starting from. I'll be showing you what the receptors actually look like, and also the mechanisms of how they work, and talk about the different functions of the different families of the GRI genes. So the four families: GRIA, GRIN, GRIC, and GRID. And I'll also be talking mechanistically about their role in learning and memory. Okay, and overall, all the way through, hopefully this will help you build up a glossary of important terms. So I guess the idea of these whole sessions is to try and give you the tools to actually listen to the scientists when we're talking to each other, when we're describing our data. And just because there are certain terminology we just use so automatically, because we've been studying it for, I've been doing this for 20 years. I think probably Dr. Swanson has probably been going slightly longer than that. In fact, I took some of his DNA out of the freezer on the very first day I arrived in my labs. So he's definitely been doing this longer than me. And the danger with that, of course, is that we end up being so familiar with our terminology, we just use it, and it's gone in a flash, and you can be like, what was that? So I'm hoping that at the end of this session, I will be able to kind of increase your knowledge of some of these useful scientific terms. And of course, all the way through, if you have any questions, feel free to stop me and ask, or save them up for the end. That's whichever way you want to do it. Okay, so just to recap, so last week, or a couple of weeks ago, you heard about DNA, which is this beautiful structure depicted here. There are 2 strands of DNA, and these 2 strands are paired. So individual bases, the individual units of DNA, pair across the double helix. Now, these DNA bases are read in groups of 3, and each group of 3 corresponds to a particular amino acid, and that's where I'm going to start talking about with the next slides. Now, a good thing about this double helix is that there are 4 bases of DNA: the A's, G's, C's, and T's, and A's and T's always pair up with each other, and G's and C's always pair up with each other. And so what that means is that there's as much information in one strand as there is in 2. So when you separate DNA, it has all the tools required that it can replicate itself. And this replication is incredible, with very, very high fidelity. There's only a mistake it's something like 0.000000001 of a time does DNA ever get it wrong and put the wrong base in there. But unfortunately, even though very rare, these mistakes in DNA replication do happen. And when that happens, it can lead to changes in amino acids and ultimately lead to defective proteins. And of course, that's what's brought us all together today somehow or other. And so in terms of the GRI genes, I'm going to talk about exactly what they do and how they work. And hopefully this will kind of give you some extra ideas about how it's possible to break a glutamate receptor, so to speak, with the improper amino acid in the wrong position. And of course, without the correct signaling that glutamate receptors are supposed to provide, this can lead to deficits in brain development, in the brain's ability to learn, and also provide, for example, damaging overactivity, which we see in the form of epileptic seizures and other kinds of excitotoxic type damage. Okay, so that was kind of the stuff on DNA that you touched on last time. Now, what I'm going to start talking about is biochemistry. Now, biochemistry is possibly not the most fearsome term that you might end up hearing today. It's quite straightforward, really. So it is the chemistry that occurs within biological systems. And whenever I hear this term, I think of this. So this is me. I didn't always used to be stressed and balding. Back when I was about 16, my dream job all the way through childhood was basically that I was going to write computer games for a living. That's what I really wanted to do. That's what I thought I'd end up doing. But I couldn't shake the feeling that I was much more interested in my science classes than I was in my computing classes. And so there was one particular topic we covered, which was DNA and protein. And this was, I think, at the start of my A levels, like I say, I was about 16. And I was just absolutely fascinated by the way that you could take the chemistry that I enjoyed studying, but in a biological system, these proteins could kind of maneuver that chemistry in 3 dimensions and just make it do fantastic, wonderful things. And so I was kind of hooked, and that was my kind of starting point when it actually comes to all this stuff. And so what do I mean by this chemistry? Well, when DNA is read in these groups of 3, there are 20 possible amino acids that can be coded by those 3 bases of DNA. And each of these amino acids has a unique size, shape, and it's got its own chemistry as well. So, and these are the 20 amino acids here. And what's very important in terms of the structure of proteins is that certain amino acids can share a particular kind of environment. So these 10 amino acids here are called non-polar in this graphic, but you can also call them hydrophobic, they don't like water, or lipophilic, they do like lipids. And in general, what this means is that these amino acids with these side chains, they don't really like being in aqueous environments. They will always try and fold away and hide themselves. And so they tend not to be on the outside of proteins, or they tend to be in the cell membrane, which is a much more permissive environment for that. By contrast, these 5 amino acids here are called polar, and or hydrophilic or lipophobic. And so these ones tend to enjoy being in a watery environment. It takes much less energy for them to be in a watery environment. And so as proteins are written, as the string of amino acid that makes a protein forms, these little bits of chemistry can all come together to help form the protein into the final shape. And of course, ultimately in the mature protein, their exact positions, they have their own little chemical groups, some of them more important than others. So for example, as well as these ones liking being with each other and out of the water, and these ones not minding being in the water, there are charged amino acids as well. And we'll probably remember from chemistry way back that like charges, opposite charges attract. So positively charged amino acids and negatively charged amino acids are likely to associate and to interact strongly. And it's worth bearing in mind that glutamate itself is actually also an amino acid that can be part of the protein. And because it has a negative charge, there's a key couple of amino acids within the ligand binding domain, the bit that binds the glutamate of the protein, that have a positive charge. And then by the negative charge of the glutamate interacting with the positive charge of the arginine, that is what binds the glutamate into the ligand binding domain. So while on a microscopic level, the chemistry, they all have their own individual chemistries. In the mature protein, they all have their own chemistry, they all have their own function. And so if anything goes wrong with these particular functions, that the right chemistry isn't in the right place, then that can lead to defective proteins. Okay, so as well as their roles in the final protein, as I alluded to, it's very important that they form the right shape. Okay, and in general, the terminology of this kind of field, this kind of science, is called protein folding. And you start off with a polypeptide chain. That's a string of amino acids that's coded by DNA, and this is the primary structure. So every amino acid is always next to the same amino acid in the string. Now, what starts to happen as the protein is making this protein, the string of amino acids, is that certain chemistries start liking each other. So if you have a couple of hydrophobic, these non-polar residues, they're going to want to share each other's space because they want to be away from the water. And as the protein begins to form, you start forming what's called secondary structures. So for example, if let's say this third amino acid here is a negative charge and this fifth amino acid has a positive charge, even though they're not right next to each other, they're going to start trying to share space because the opposite charges attract. And that starts to form little bits of secondary structure. Now, when you actually start looking at the detailed cartoons of structures, you'll probably notice there's quite a few spirals in that structure. And this structure here is called an alpha helix. And it's quite a good and stable way of a protein kind of going from A to B in a controlled way. And we particularly see these structures when proteins are passing through the cell membrane. And so I'll show you some of those a bit later on. And so primary is amino acids, secondary is how they locally interact with each other. And the tertiary structure is also very important for glutamate receptors, because certain chunks of the protein that are nowhere near each other in the coding sequence belong with each other in the final subunit. And this is called tertiary structure, where something that's quite near the start of the protein and something quite near the end of the protein nonetheless come together to form the structure. Okay, and then finally we have quaternary structure, which is where entirely different proteins during assembly can come together. And the quaternary structure of the glutamate receptors is what I'm about to show you right now. So we're very, very fortunate over the last, since 2009, we've understood more and more about the three-dimensional structures of glutamate receptors as they form and as they work. And so even now, as of the most recent one with the grids, we now have three-dimensional structures for all four of the main families. So the Ampa Receptors formed by GRI genes, Glutamate receptors are from GRIC, NMDA receptors are from GRIN, and GLUD receptors, the delta receptors, are from GRID. Now, as you can see, looking along the line, they're all subtly different, but they've all got lots of similarities. And this is generally what happens when you have proteins that are in related families. They form in general the same shape, but there's certain key differences. So they come together as a tetramer. There's a blue, a yellow, a red, and a green subunit forming this tetramer. And in terms of Ampa receptors, they're mostly what we call heteromers. So these are combinations of proteins that aren't identical. So there's 4 different Ampa receptor genes, and a very common combination of Ampa receptors, for example, has 2 GluA1 subunits and 2 GluA2 subunits. So they come together in a mix. But they can also form only on their own, like GluA1 homomers, where every subunit is GluA. Similar kind of assembly rules apply to kainate receptors. And for NMDA receptors, on the other hand, they can't form homomeric receptors. They have to form a combination of different subunits. And then finally, the delta subunits come together. These, it's kind of less clear, less work has been done on these, but it seems likely that they can only form homomers. So if you have a GLUD receptor, it's either all 4 subunits of GLUD1 or all 4 subunits of GLUD2. And just another general note about the structure, they all have these transmembrane regions. That's where the receptor is anchored in the membrane. And this is where you see a lot of these alpha helix structures. It's a very stable way of passing through a membrane. And in the middle of this is where the ion channel is formed, and that's where the electrical currents that the glutamate receptors pass. That's where they're. Okay. So that's a kind of the beautiful structures that structural biologists can give us. This is these are the kind of cartoons that scientists like me who can't really draw that well will give you. But hopefully it kind of simplifies it a little bit. So I've taken it, this isn't a tetramer, this is just half the receptor in effect, but it allows me to show you the key mechanisms of how they actually produce electrical currents, how they actually work. So in general, the structures get broken down into 4 main groups. So the four main modules or domains. So the amino terminal domain that's here towards the outside, the ligand binding domain that's just to the outside of the membrane and can bind glutamate, but also some of the subunits also bind glycine. So the GluN1 NMDA receptor and the GluD receptors also can bind glycine or D-serine. There's the transmembrane region, which forms the pore, and also the C-terminal domain, which is the kind of the bits that just stick inside the cell. And as well as these four main domains, there's also it kind of falls in between. There's these linkers that link the ligand binding domain to the pore, and these are absolutely crucial in terms of the way the receptor works. So we mustn't forget about them. And the reason and the mechanism by which the linkers are so important is as follows. So if you imagine a receptor with no molecules bound, no agonist bound, sitting in the membrane, when the agonist binds, by which I mean the molecule that closes it, which is glutamate or glycine, depending on the receptor, what that means is that the ligand binding domain, this central region here, it has a structure that often gets referred to as a clamshell. So you have something at the top and something at the bottom, and then glutamate comes in the middle, and then the ligand binding domain closes around it. And once it closes around glutamate, that means that this lower part of the ligand binding domain moves up, and that pulls on these linkers that actually pulls open the pore. So it's a beautiful little machine almost. You bind glutamate, it closes it, it grabs the glutamate, and in doing so, it actually pulls open the ion chain. And it's this arrow here that depicts the little electrical currents that the glutamate receptors produce. Okay, and as well as obviously the forward process is that this is what opens the receptors. It's also very important that they can close as well. There's a couple of different processes for this: deactivation or desensitization, which you might hear us talk about, but which are basically just the receptor closing. And if there is a variant in the receptor that disturbs this this whole process, even quite in quite a minor way, that can lead to GRI disorder. So, for example, if you have something that increases the affinity of a receptor, it will hold on to the glutamate for too long. Or if you destabilize the gate, maybe it'll come open too much. Or if you stabilize it, maybe it'll stay closed too much. And any of these things are possible to actually really hugely disrupt the way the brain is supposed to work. Okay, so yeah, so this is really already covered by Megan, but I'll just quickly go over it again so I'm saying what I wanted to say, which is that the thing to know about signaling in the brain is that the signals that travel along neurons are broadly they are electrical, whereas the signals that pass between neurons are chemical. And the glutamate receptors act at the point where the chemical signal is turned back into an electrical signal. So they're vital for, if neuron 1 wants to talk to neuron 2, it needs glutamate receptors to get that signal across. And so they really are super important for the way the brain works. 86 trillion synapses, was it? I can't, maybe I might have a decimal point wrong there, but yes, this is happening all over the brain the whole time. As you're listening to me talk, you are releasing trillions and trillions of molecules of glutamate, just even to process even a syllable of what I'm saying. Okay, and in terms of the actual structure of neurons, they have the cell body here, and kind of upstream of that from the synapse, you have processes called dendrites, and these are the parts of the neuron that catch all the signals. The cell body or the soma helps to process all those signals. And then there tends to be a single electrical output called an axon that then, if enough excitation has happened, they will pass on the signal. And so just kind of zooming in a little bit more into an actual synapse here, the presynaptic side, so this is from neuron 1, electrical signal passes down here, and if electric signal makes it to the synapse, it triggers glutamate to be released. Okay, and glutamate normally lives in these little packets called vesicles. And when the signal comes in, these vesicles fuse with the membrane and just release a little puff of glutamate out across the synapse. And it's on the postsynaptic neuron, so neuron 2, where the glutamate receptors sit, and they catch these glutamate signals and turn them back into electricity. And if there's a large enough signal through the glutamate receptors, that then means the second neuron is more likely to pass on the signal. Okay, so the brain only wants to pass on signals when it really needs to. Okay, so I mean, I could talk all day about glutamate receptors, but I'm going to focus on, and Dr. Swanson will have to forgive me because I'm kind of glossing over kainate receptors a little bit in kind of what I'm going to talk about from now on. But Ampa receptors and NMDA receptors work together. They have a real symbiosis. So, and they look very different. They behave very differently. So Ampa receptors bind glutamate with a very low affinity. That means they can only really bind a lot of glutamate when there's a very high concentration of glutamate. But the good thing about this is that because it's got low affinity, it lets go of the glutamate very quickly as well. And that's really important how Ampa receptors work. They give this really short signal, and this allows them to act at very high frequency. So this is the job of an Ampa receptor, is to produce these really fast signals, which can be at high frequency, they can be at low frequency, but that's how they work. NMDA receptors, on the other hand, bind glutamate with much higher affinity, and this gives a much longer signal. So Dr. Swanson, I'm sure, will talk in more detail about this later, but you end up with signals that look like this. So as time progresses, going from left to right and downwards is the electrical signal. Ampa receptors, glutamate's released from a vesicle. They open really, really quickly because there's that high concentration, but very quickly the glutamate drifts away and they very quickly close. NMDA receptors, on the other hand, are much slower, and so when they open, they produce a bigger signal. However, they don't normally, they have to be in the right conditions before they can pass a signal. So the pore of NMDA receptors is normally blocked by an ion called magnesium. I'll come on to this in a bit more detail shortly. But what happens is if there's enough Ampa receptor activity, this relieves the block of magnesium from NMDA receptors. And so as long as there's enough Ampa receptor activity, you also see NMDA receptor activity. So they really work very, very closely together. Okay, so far I've mainly focused on the central parts of the channel, so the pore and the ligand binding domain, but the amino terminal domain and the C-terminal domain are also very important for Ampa receptors. You could argue they're slightly less important in the sense that they tend to contain less disease-causing variants. However, there are nonetheless there are patients with variants located in this outer region and in the C-terminal region. So important things the amino terminal does, it helps to build the receptor. So in terms of when the quaternary structure is first being formed, it helps to bring two different subunits together, and then the receptor can actually build itself. But at least in terms of the GRIN and the GRID genes, they can also have their functional properties modified by the immunoterminal domain. So there's things that will bind to the immunoterminal domain that affect the rest of the channel. And also of great importance for the immunoterminal domain and the C-terminal domain is that they help the neuron, they help the receptor be where it needs to be. Because neurons are very beautiful, very complicated cells, but they're still cells nonetheless. And actually only a tiny, tiny fraction of the surface of the cell is actually synapse. And so you really need to make sure the glutamate receptors get to where they're going to be. Otherwise, they can't be activated by glutamate if they're not at a synapse. And the amino terminal domain and the C-terminal domain of the receptors is very important to help the synapse kind of catch the receptor, so to speak, to make sure that the signals are passed on. And I guess those factors about receptors being in the right place at the right time is no more important than during brain development. So the majority of stuff I've talked about so far is actually only focusing on this small aspect of what glutamate receptors do in the mature brain. But as you can see, if you kind of look for your gene of most interest in the list here, you'll see that virtually all of the glutamate receptor genes are important for various things around the adult brain, but also in the developing brain as well. And this is important because if something goes wrong with the glutamate receptors, this can mean that the effects of this are felt very early in development. So neurons start as progenitor cells, stem cells, as all of the cells in our body do. And then as the brain develops and they grow, they start to grow processes, they start to move around in the brain to find their final place until they ultimately become adult cells. And one of the key things that they need to do this, to navigate their way in the brain, is to have a population of receptors on the surface of the cell that will allow them to be guided into the right place and to make sure they know they're in the right place. And glutamate receptors are included in that body of receptors that are very important. And so if you have, for example, a loss of function variant in one of the glutamate receptor genes, it can make developing neurons less able to detect their environment. Or equally with gain of function, again, they get the wrong signal. And when you think about going from just a handful of cells that you are when you're a few days old to having a brain with trillions of neurons, all supposed to be in the right direction, if you start messing up the process early on, then it can be very, very damaging. And so I just want to finish by talking about one particular aspect of glutamate receptor physiology, which is really important, which is their role in learning and memory. So modification of glutamate receptors is crucial for what we call synaptic plasticity. And what that means is it's the way that synapses can adapt to the signals that they receive. And so when they undergo particular patterns of activity, they change their behavior accordingly. So, and this process is certainly responsible for memories that can form over a few seconds and last for hours and days, but even memories that we remember from years and years later. So if you think about, just randomly think of some class of yours at school and think of the person you sat next to or the person next to you on the register. Even you may very easily be able to come up with the name of someone you've never even thought of their name or thought of their face in 40 years, but all of a sudden you can still remember. And so the initial process of laying down those memories is absolutely totally dependent on glutamate receptor activity. And so it's something that is very widely studied and of great interest. And the main mechanism of learning and memory formation that we tend to use as scientists is looking at the behavior of Ampa receptors and NMDA receptors in the hippocampus. And this is a part of the brain that is very, very important for forming memories. And it's also, in terms of disease, it's actually the center of the majority of epilepsies that can occur. And so it's very, very important to understand glutamate receptors. And in terms of how this memory actually happens, again, this is another example of that vital symbiosis between Ampa and NMDA receptors. So something else that Megan mentioned is that you use 20% of your energy is used by your brain. When you're sat there doing nothing, your brain uses more energy than anything else. And the majority of that energy is spent pumping positively charged ions out of your neurons. And in general, what this means is that the inside of your neurons has a negative charge. Okay, and so you end up with lots of positive ions outside, and it's quite negative inside. So, and that almost acts like a bit of a battery for the brain. It kind of builds up some chemical energy that can be released whenever needed. And of course, it's the glutamate receptors that cause that energy to be released. Okay, so at rest, there tends to be enough glycine around to gate NMDA receptors in general. Obviously, it can fluctuate, but as a general rule, we can assume that there's enough glycine around to gate the receptors. Now, when a weak glutamate signal comes along, that would be enough to put glutamate onto Ampa receptors, and they open. And when they do this, they pass an electrical signal. The inside becomes a bit less negative. But as I mentioned, NMDA receptors are blocked by magnesium ions. And so even though now they're occupied by glutamate and the channel is open, it's plugged by magnesium. So you can't do anything about that. And there's no NMDA receptor current. However, if you get a much stronger Ampa receptor signal, many more Ampa receptors are activated, the inside starts to become more positive. And when the inside becomes more positive, it pushes that magnesium away to the outside. And this allows NMDA receptors to open, and they can let calcium through. Okay? And it's this calcium signal which mediates the majority of the synaptic plasticity that we learn about. And so all of this process of learning and memory is completely reliant on Ampa receptors and NMDA receptors kind of living together and working together. Okay, and then just to kind of visualize exactly what that means, well, if you start off with Ampa receptors and NMDA receptors in a synapse and they get this calcium signal, one of the things that can happen in the very short term is that the signal passing through an individual Ampa receptor can be increased. Okay, and so when you try and stimulate the same synapse, 10 seconds later, 30 seconds later, the signal is already bigger because you've modified the receptors that are there. A bit kind of on the same kind of the seconds to minutes timescale. Then you also increase the number of Ampa receptors that are there. So again, this signal gets bigger. And then finally, on a slightly longer time scale, the synapses themselves begin to change shape. So they actually become much, much bigger. And that allows not only more Ampa receptors to live there, but also eventually you upregulate NMDA receptors as well. And so all of this comes from high degrees of Ampa receptor activity, allowing NMDA receptors to be activated, which then produces these changes in the synapse. And not all plasticity is positive. There is also negative plasticity as well, where you start to lose synapses. So if a synapse doesn't get stimulated for a long time, or there's just a persistent low level of activity, the neuron thinks, okay, we don't need that synapse anymore, and it actually removes Ampa receptors from it, and then ultimately this can lead to loss of synapses. Now, this is part of normal learning and memory, but it can also be a part of neurodegeneration as well, and when things start to go wrong with the brain, Ampa receptors are lost, and then ultimately the synapses are lost as well. Okay, so just to, I've kind of done a kind of a glossary as well as rather than a traditional summary for this talk. So these are all kind of terms that I've raised in this talk, and you may well come across again when you listen to us in general talking about the work that we do. So I'll just quickly run through it now. But so amino acids are the single building blocks of the protein that are coded by 3 bases of DNA. The quaternary structure is how multiple proteins can come together in a complex. Heteromers, now this means a protein complex made up of different subunits. So for example, NMDA receptors are always heteromers. And all of the glutamate receptors are tetramers, which means they're made up of 4 subunits. Ligand binding domain binds glutamate, which allows the pore to open. And that is done that's performed by the channel gate, which is the central part within the membrane of the receptor that allows ions to pass. There's also very important linkers that link these 2 domains and allow the movement of the ligand binding domain to open the pore. And then in terms of neurons, we have synapses, and these are these chemical junctions between neurons where glutamate receptors live. Dendrites, which are the part of the neuron that receive the signals and pass them down to the cell body. And then axons, which are the part of the neuron that release the signal. And then finally, we've got magnesium, which blocks NMDA receptors. And when that's removed, they let an awful lot of calcium through, which can allow memories to be formed. And in general, we call this adaptation of synapses synaptic plasticity. Okay, thank you very much for your attention. I'm happy to take any questions now or after the second talk. I'll be here the whole time.
Thank you so much, Dr. Coombs. Dr. Swanson, if you want to go ahead and share your screen. In the meantime, do we have any questions from the audience? Okay, there will be time for questions at the end.
Okay, can you see my screen?
Yes.
Okay, well, thanks for inviting me to talk this morning. I'm going to maybe take a step back a little bit from the material that Ian just covered and talk to you more about how we measure those currents, how we generate those traces that you saw on the screen, and no doubt that you'll see quite a bit more in the meeting later in April. So I wanted hopefully give you a little bit of understanding how we get to those currents. Those are current traces, and how those are generated, and then a little bit about how we define gain of function and loss of function variance, and to what extent that can inform what's happening in the brain in the presence of those variants. And then lastly, I'm just going to say a quick word about how these different types of variants shape our thinking about possible therapies. Sorry about that. I guess I paused that. Okay. So just a reminder, Ian just mentioned this, but I wanted to make sure that I reiterated. So we talk about genes, and these are the GRI genes that you all are familiar with. They make the proteins, the GLU, A, D, K, or N proteins, and we refer to those in the receptor form as AMPA, delta, kainate, or NMDA receptors. And I'm going to talk today about kainate receptors, but I'm using these just as an example of general GRI behavior and analysis. So nothing I'm saying is particularly unique to kainate receptors. It's just that those are the examples I'm going to give. And so what you've seen in Ian's talk, what you've probably seen before in research presentations, are traces that look a lot like this. So you oftentimes will see a line, and it'll be going down, and then it'll go back to the original starting point. And then we'll call some types of traces control traces, and some of them we might call gain of function traces or receptors or variants. Some will have complex effects that are difficult to define in a simple term. And then we'll talk about loss of function. And what I think everyone can appreciate is that these all look generally the same to, as a first approximation. As people who've worked in the glutamate receptor field, Ian and I can look at these traces and we can think, what is the protein doing when, for example, the trace is going down? What does that mean in terms of the receptor? And then how might the receptor be operating differently when you see a trace like this second one in red? And so we've been trained to connect these lines, these current traces, to what receptors are doing and interpret that information in the context of how variants might affect the function. And we generate graphs that look sometimes like this, and sometimes they come in other forms. And what I hope to do is to give you a little bit of background information so that when you see these kinds of traces in the meeting or in research presentations, you kind of have a sense of what they mean and how we are thinking about their meaning in the context of the receptors. And the way we typically generate these traces and the way we analyze the different function of glutamate receptors, these are ion channels, so we can measure electrical currents through most of them. The technique that we use is a technique called patch clamp or voltage clamp electrophysiology, and I'll tell you what that means or what that approach looks like. And this is an approach that is, there's a couple variations on this approach, and it's used very commonly to characterize and analyze glutamate receptor currents. And the strength of the approach is that you can measure very, very small currents. It's very powerful. There's not a lot of current going through these channels in a sort of general sense, but we have machinery that will allow us to measure those currents with high fidelity. We can think about how large they are, how much current is passing, and we can think about how long that current is passing for, and understand from those parameters something about how the receptor operates and how it might operate differently when it has a variant in it. And then there are weaknesses as well to this approach. It's slow, it takes time to do this analysis, and it can't tell us anything about variants that don't actually change the function of the receptor. Those variants are essentially invisible in a functional sense to us. And then lastly, I'm going to maybe spend a little bit more time on this last point, and that is that glutamate receptors in neurons, in those synapses that Ian spoke about, they're typically much more complex than what we can analyze what these traces here represent. And so that also introduces a limitation in what we can infer from these kinds of traces, these currents. Okay, so these are not currents in neurons, these are currents in reconstituted systems. And so you might ask yourself, well, why don't we just study the currents of glutamate receptor function in neurons? And that turns out to be quite challenging because the receptors in neurons in the brain are really quite complex. They are made up of the 4 subunit proteins that Ian mentioned, tetramers, and those change. They change during development. They change in different brain regions. They're different in different brain regions. Even in neurons within a single brain region, like the hippocampus, they are different in composition. And even within individual neurons, they differ in their composition. I just want to give you one example in that part of the brain that Ian mentioned that's important for learning and memory, the hippocampus. This is a mouse hippocampus in red, looks vaguely like a banana. If you look at the individual layers, so these dark purple layers here are where the cell bodies lay. And we named different parts of the hippocampus with different names, CA1, CA2, CA3. If you just look at one neuron in the CA3 region, it looks like this beautiful neuron here on the right side. And this is called the hippocampal CA3 pyramidal neuron. It's just one example of all the neurons that line up in this dark purple area. So one level of, or two levels of complexity in these neurons is first, again, referring to kainate receptors, is that just the initial part of the dendrites here actually have kainate receptors. So there are ways in which the cell controls where the kainate receptors are placed in neurons. So they're only placed in this first part and not in all the more distant parts of the dendritic tree. And even within this this sort of limited zone in the dendrites, you have quite a large diversity of receptor subunits that are combining into that 4 subunit receptor. So of the 5 kainate receptor subunits, you actually have 4 of those combining in different ways into different receptors. So you can appreciate how complex analyzing these receptors might be. And here I've also put NETA1, which is a protein that assembles that is part of the kainate receptor complex, but is not part of the main channel that Ian described. Okay, so it's too complex in essence to study these receptors in a defined way in the brain. And so that's why we try to reduce that complexity. And we do that by analyzing them outside of the brain in what's called in vitro systems or expression systems, where we can put in DNA and have those cells express billions of receptors for us to analyze effectively in electrophysiological analyses. And so these cells are a lot less complex than neurons. They don't have all those branches that you saw in the cartoon and the picture I just showed you. They're relatively small, which is advantageous for physiological analysis. And they don't have different brain regions or neuronal regions that they are expressed in. It's just basically a bag of a cell, and so it's just being expressed on the cell membrane. And then lastly, it allows us to control the receptor composition. That is, we can try and put in specific subunits to make up those receptors and then analyze receptors that have a known composition of subunits. The limitation of those expression systems is that the receptors are obviously not where they're normally supposed to be in neurons or as synapses. So that is, then there are certain aspects of being in neurons that are not reproduced in these expression systems. And then we can simulate the receptor combinations that are found in the brain, but almost always those are too simple relative to what appears in the neurons. How do we go about analyzing these receptors that we express in neurons? So essentially we introduce DNA or RNA into cells using very standardized techniques, molecular biology techniques. Those cells have the ability to then use that cDNA or RNA to synthesize, as I said, billions of receptors, and they move them out onto the cell membrane where we can do our electrophysiology or biochemical analyses. And I'm going to focus mostly on electrophysiology. So what does that look in actuality? There's really 2 main systems that we use for expressing loads of receptors. They're either Xenopus oocytes, which seems like a very strange sort of system to use, but it has some advantages. They have very large eggs that if you inject RNA into them, they have all the machinery to make those receptors, and then they'll express them on the membrane of those eggs. And you can record them with a technique called 2-electrode voltage clamping. You can also use mammalian cell lines. So the cell line of choice is often referred to as an HEK cell. And what you do here is you again, put your cDNA or your DNA expressing your receptor subunit into a dish, put in some what is known as transfection reagent, just basically a chemical that's optimized for getting these DNAs into the cells. And those cells express loads of receptors, again, on the cell surface. And I think that you can appreciate these are much less complex cells than neurons. So the general objective here is to measure currents through glutamate receptors. They're ion channels, so ions go through them, and in doing so across the cell membrane, they generate a current. A couple of terms, again, I want to reiterate what Ian said. We use a lot of jargon, and we're aware of it, but sometimes we slip and say things that mean nothing to anybody else, except for people who've been doing this for a long time and studying electrophysiological current recordings. So one thing that I've already said, but not defined, is patch, as in patch clamp recording. And so a patch in this sense just refers to a little bit of membrane that is in contact with a glass electrode, what we use to measure those currents. And it has to contain at least one channel, or typically contains many more channels to be of interest and to be of use in our physiological analysis. And clamp, the word clamp in this case refers to a mechanism for controlling one of the experimental parameters in our patch clamp recording. And that's achieved with a piece of equipment called a patch clamp amplifier. Now, I'm going to apologize in advance. There's just a little bit of math here, and I think hopefully this will be clear and give you a sense of how those currents are generated and measured. So at the heart of electrophysiological analysis is this very simple relationship between voltage, V, current, I, and resistance, R, and the inverse of resistance is conductance, G. And these are defined here. And so why is this important to tell you? Well, it tells you how we end up measuring currents through glutamate receptors. The clamp part of patch clamp or voltage clamp recording is the voltage. So our amplifier, our piece of equipment, holds that voltage stable. So we are controlling the V part of this equation. The current is what we measure. That's also measured by our patch clamp amplifier. And then the resistance or the conductance, that is something that's an intrinsic property of the glutamate receptor. So what we're actually trying to infer is something about the receptor, and then how those properties of the receptor change when there's a variant introduced. So we're measuring the current. Those are the downward traces that you saw, and then inferring something about the receptor based on the resistance. Okay, so what does it look like sort of more practically? These are Hek cells or HEK cells. We grow them on a dish. They don't look like spheres here, but they do when you record them. They're quite easy to grow, and they're generally pretty happy cells. And we bring a glass electrode. So this glass electrode here on the right side, that's connected to the patch clamp amplifier that I mentioned was going to control the voltage and measure the current. Our receptors are expressed on the membrane of these HEK cells, the target cells. So we use a very, very fine control manipulator that's moving this glass electrode down, and we go down and we touch the cell membrane, and we form what's called a high resistance seal. So it's just the contact between the membrane and the glass electrode is so tight that any currents we measure predominantly are coming through glutamate receptors and not through other sources in the cell itself. And then if you blew it up and looked in cartoon form, this is where the tip, where the glass meets the cell membrane. Cell membrane is red here. The glass is this white structure here. And what we typically do is we rupture the membrane where the glass electrode is connected. And so then the inside of the electrode becomes contiguous with the inside of the cell. And that's what allows us to control the voltage of the cell across this red membrane. And so our little glutamate receptors are these green structures right here. And that's what we want to measure. We want to measure the current that goes through those receptors. We're controlling the voltage across the membrane with the amplifier. And then we're inferring something about the resistance or the conductance of the channels in order to understand how variants cause changes in those channels. Right, that's what you said. Okay, this is what a patch clamp setup looks like. This is because I'm on the older side. This is an older setup. Ian probably has a newer, fancier setup to show you. But what I can point out here is this is the main part of the patch clamp amplifier. We have a microscope. Obviously, these cells are very, very small, so we need a high power microscope to even see them. And the cells are sitting here on this silver platform here. They're sitting right under this little brown cylindrical object, which is the objective. That's where the light is focused, and it's focused down on cells that are sitting right in a little stage here. And over here, it's obscured a little bit, but this black piece of machinery is the micro manipulator. So that's what allows us to move the electrode at a micron level. So we can really precisely target those receptor expressing cells. Over here is the patch clamp amplifier that I've mentioned several times. So this amplifier has a wire that comes out the back through the copper cage and then up. So you can see it going up here, and it's going up to the manipulator area, and it's attached to our electrode. And then through this process of recording with all this machinery, we generate glutamate receptor currents, which on our computer look a lot like this. You can see there are downward deflections. Those are the channels opening, sodium flowing in. We measure that as a current on our computer, and then think about what that means about the receptor function. Okay, so back to our one of the original traces, or something that looks a lot like it. So they look like this. The channel at rest is where this, what we call baseline, is located. So essentially the channels are not open at this point. They do open when you put glutamate on them. When you expose the cell to glutamate, loads of channels open, and you get a current through those channels. And so what's happening during that current is typically a bunch of sodium ions are flowing into the cell through the channel. And that's what we measure as a current. Glutamate receptors are not designed to stay open for very long, so they open quickly and they close quickly on the order of milliseconds. And so that's what's happening when this current goes back towards baseline. Sodium flow is decreasing because the channels are closing. And then when you take glutamate off entirely, they completely go back to baseline because glutamate goes away and the channels close. So in sort of at the level of the protein, it looks kind of like this. Is at a relatively slow pace, so I apologize for this, but it's leisurely, the receptor operating as though we're at the beach or something. So that's the glutamate flying into the ligand binding domain that Ian mentioned. And what I hope you can appreciate is there's this large protein on the protein scale. There's a pretty big protein, pretty big receptor. The whole thing is sort of moving around, and all these conformational changes are designed to do one thing, and that's open the pore, which is located down here. So the membrane would be here. The ions would flow in through the middle of these alpha helix structures that Ian mentioned. And that's when we generate the current. And then at the end, where glutamate is taken off, glutamate flies away and the channels close. So that was, like I said, a leisurely process, but in fact, this happens about 10,000 times faster under normal conditions. So we do all those measurements, create those currents that you saw, and we generate tables that look like this. So they're parameters that we can measure about the currents that tell us something about the proteins. Parameters like deactivation, desensitization, and lots of other things. And it's these values that we use to think about how 3 variants might change the function of the receptors. And then back to these original traces. So a bunch of traces. So these alanine 657 to threonine, threonine 660 to lysine, these are variants that we've introduced to what we call recombinant receptor subunits that mimic variants that are found in kids with GRIC, de novo GRIC changes. And so we can look at these. Here we've put on glutamate for a very, very short time, only about a millisecond. And here we put it on a slightly longer time. And we do that to capture 2 of those parameters that I mentioned before, deactivation or desensitization. And we generate these values just by looking at how fast the current decays back to baseline. So it would be we fit this to a mathematical equation as it goes back to the baseline. And what I think you can appreciate is this one goes back very fast. This is the glutamate, this is the GluK2 or the GRIC2 gene that is unmodified, that doesn't have a de novo variant in it. These are mimicking de novo variants. And you can see how the current changes, the time course of the current changes. This one gets quite slow, and as does this one, and does the T660R. And then conversely, the isoleucine 668 threonine gets very fast, even faster than the unmodified GluK2. And so what we can infer from these then is, or we can categorize these as gain of function, loss of function, and complex effects. So we would categorize this A657T as a gain of function. And that's because if you compare this deactivation time course, let's say it's 2.5 milliseconds, compared to the variant 657 variant, you can see this variant is really slow. So maybe 10 times slower. What that means is more current passes through that receptor, more sodium ions passes through that receptor each time it gets activated compared to the unmodified receptor. So that's why we call it a gain of function. It's also more sensitive to glutamate. If you think about what Ian said, that Ampa receptors and kainate receptors are relatively low affinity, typically they require about 10 times higher concentrations of glutamate than NMDA receptors. Well, these receptors require much lower concentrations to be activated. Loss of function is fairly straightforward to define also. So these get faster, less current flows through for a given activation. There are also fewer receptors on the cell surface. That's not reflected in these numbers, but by other measures that we can make. And they're less sensitive to glutamate. They require higher concentrations to be activated and have current flow. And then there are variants that defy easy categorization, which I call complex effects. So for these 2 types of variants, you have much slower current, so you might call that a gain of function, but they also have much less current passing through the receptor when they're activated. So that actually seems more like a loss of function. So they have 2 different or multiple different effects. And so that we can't really call them a gain or loss of function. They're somewhere in the middle. So that's how we use these reduced systems with reduced complexity to categorize gain of function, loss of function, or just more generally look at how variants change the operation of these receptors. But there are limitations. So what I just showed you is a comparison between a homomeric GluK2, unmodified GluK2. Homomeric means each of the subunits, it's hard to tell, but there are 4 subunits in this tetrameric receptor as normal. All of these contain exactly the same subunit, and that's a GluK2 subunit. If we then look at one of the variants I showed you, all the receptors have exactly the same subunit in each of the 4, or each of the 4 subunits are the same. This is a useful way to compare the properties, the functional properties of the receptor, but it's not what happens in real life in the brain. So it's a super simplified version of what these receptors are normally, how they're normally operating. In fact, because de novo variants only occur on one of the two alleles for GRIC or GRIA or GRIN genes, what we expect in the brain is that the receptors themselves would be a mixture of unmodified GluK2 and modified with de novo variants of GluK2. So you get kind of a combination. So half would be the variant, half would be unmodified as a best guess, as a first approximation. But even this is too simplified because, as Ian said, these are assembled with different subunits typically. So one of those subunits for KNE receptors is the GluK5 subunit. It's a very common combination, GluK2 and GluK5. So if we think about then what's happening in the brain of an individual with de novo variant and GRIC2, it might look something like this, where you have one variant version of GluK2, you have one unmodified version of GluK2, and then you have a couple of these GluK5 subunits, which are not modified either, which will, however, change the properties of the receptor. That's also too simple, though, if we're thinking about what's going on in the brain. Because, as I said, they have these proteins that bind to the receptor complex and modify the functional properties called netoproteins. And for Ampa receptors or GRIA, they have a whole bunch. They have many different types of these, what are known as auxiliary proteins. And you can see in purple here, these also bind, and through that interaction, modify the properties of the receptor. So now, when you get to this level of complexity, the GluK2 with the variant is one part of a 4-subunit tetrameric receptor plus auxiliary subunits bound. So it's part of a complex. So the comparisons we make here on the far left side are really the simplest version of what we understand the de novo variants to be doing to the receptor. But in fact, as we build this receptor closer to what is actually present in the brain, it becomes quite complex and more challenging to predict the functional effects and what's actually happening when the variant is present in the brain of individuals. But it's nonetheless, it's really the best way we have of predicting what's going on in the brain and predicting how these variants might affect the receptor function and the neuronal function and development as well. So it's both a useful way of approaching the analysis of glutamate receptor variants, but we always have to keep in mind there are limitations to what we can learn about the receptors in neurons. And as Ian mentioned, there are variants that occur in glutamate receptors or GRI genes that don't have any functional effect. And so what that means is in our patch clamp physiology analysis, they're essentially invisible. They look just like the unmodified receptor, even though they are damaging variants. And that can occur, for example, in GRIA or GRIC genes in particular, if those variants occur in the N-terminal domain, this outermost domain. That doesn't mean they're not important or that they don't have any real effect on the receptor. They do. It's just that we can't see it in our patch clamp recording analysis. Similarly, what these structures leave out is a whole bunch of protein on the inside of the cell. The way of resolving these structures is not able to resolve the inside part of the protein, but they also contain damaging variants, although in lesser numbers, as Ian mentioned. So why might this occur? Like, what's happening here? Well, essentially, I'm going to go back to the biochemical processes that Ian mentioned. If we look at a neuron here, it has its dendrites, and here's the cell body, the main cell body. And this is meant to be an axon, but it's very huge. So where the receptor gets made, assembled, folded, those processes at the very earliest stage are located primarily here in the main cell body. And you can think of that as a process similar to what happens in a receptor factory, or an assembly plant. You're putting these receptors together, you know, sort of fabricating them and assembling them into their proper configuration. And much like occurs at this factory here, which is a real factory that is about 2 miles away from my house and makes Mars candy bars. And then they get transported or trafficked to their sites of action. They get loaded onto as cargo onto cellular trucks, and they get moved out along this dendrite. But then they still have to get targeted to those synapses that Ian mentioned, sites of communication. And we can think of that as just delivery to, you know, you get your product when you enter an order online, and it shows up at your door 1 to 5 days later, and that's the process of targeting those receptors. So all of these processes depend critically on other proteins interacting and shaping the way the glutamate receptors get made and get moved and get targeted. So all those variants that affect these processes are more difficult for us to detect in patch clamp analyses. And sort of the last thing I wanted to say, that I was asked to say something about how the gain of function, loss of function, complex function effects might influence how we think about therapy. And so it's challenging. As I said, the main challenge in the first place is that we can only predict how the receptors operate differently in the brain based on a reduced system. So it has the limitations I mentioned. The second difficulty is that in a general sense, and I think you got this from Ian's talk, is we can think about the effects of variants in 2 broad categories. Those are the developmental effects at the very earliest stages, how cells proliferate, how they start to form connections with each other. And the functional changes we measure don't really tell us very much about those processes. It's hard for us to infer what's happening at the level of a glutamate receptor or a cell or an axon as it travels along, based on what we can measure in our assays. That's certainly true for GRIA, GRIC. There is some information one can derive for the GRIN and MDA receptors, because their role in these earliest processes are much better understood than Ampa and kainate receptors. So where it could have some sort of informational value is in how the signaling in a mature brain might be different from unmodified or receptors that are not modified. And so we can think about how those functional changes might guide therapeutic approaches. Again, between the different categories, I think there are the ability or the confidence with which we can predict or think about therapeutic approaches varies. And so at the first level, we probably have the highest confidence for variants that occur in genes where the protein is expressed in many different places in the brain. Like it's very common, it's a common constituent of glutamate receptors in the brain. Notwithstanding Dr. Ramsey's and Dr. Collinger's effects or efforts in research, in my view, a complete loss of function variant is actually the most challenging to think about how we approach it therapeutically, because in losing function, we don't have the ability to tweak its function because it's just simply not being expressed, and so we have to replace the receptor in some way, and that can be done in mice. It's harder to think about how it would be done in individuals with a loss of function mutation. Gain of function and partial loss of function are probably the most approachable because we do have some drugs that will up-regulate or down-regulate the operation of some of the GRI receptors, particularly Ampa receptors and NMDA receptors. And I expect that you'll hear more about the GRIN modulators from Dr. Trainellis and other folks working on those receptors. And then in terms of personalized research or therapeutics, I think in terms of research, I've given you a sense of how we analyze gain of function or categorize gain of function, loss of function. So that's pretty straightforward, but the therapeutics are more challenging to think about. And the primary limitation is simply that we don't have enough drugs that discriminate between different types of receptors and receptors with different types of variants. So it's not so much that we don't understand what might be happening, although that is a factor, but it's just that we don't have a great toolbox of drugs to help target those receptors or discriminate between different types of receptors. Again, this is mostly true for Ampa and kainate receptors. There are effective drugs that discriminate between different types of NMDA receptors. So again, there's little variation between the different families. And then in my opinion, I think to really get at personalized therapy, gene editing is probably going to be the most effective way, but of course that's has its own challenges and still is in the process of being developed for use in people. And just as a caveat, these are my opinions, these are not facts, so do with them what you will. And but yeah, this is the way that I'm thinking about potential therapeutics. Okay, that's it for me. Happy to entertain any questions if you have them.
Thank you so much, Dr. Swanson. That was great. We gave you a tough challenge with explaining electrophysiology, and I think you did a great job.
Oh, thanks very much.
All right. So it looks like we have a few questions in the chat. I think Ian is starting to address one of them. That question was about what EC50 or IC50 means on a report. A parent said that the results are within ratio. So there's like GLUN1 wild type, GLUN2A wild type relative to GLUN1 wild type, GLUN2A with a variant, which is in 648S. And they wanted to get a better idea of how to interpret that.
Yeah, so I guess, so I think I already answered this, but I'll say it again. So the EC50, the E is excitatory. So that's like the molecules that activate the receptor. The IC50 is for inhibitory. So those are molecules that block the receptor. And in general, the lower those values are, the higher affinity, so the more tightly things bind. So whereas Ampa receptors, that doesn't sound good, that's something related to a pet, I imagine. Whereas Ampa receptors have a low affinity, so their EC50 would be very high. NMDA receptors have a higher affinity, so their EC50 would be lower. In terms of the exact IC50 they would have looked at, I'm not quite sure. I'm not quite sure exactly which drug. In terms of the presenting it as a ratio, I think that's important because, you know, you can say the EC50 is 1 millimolar, or you can say the EC50 is 1 micromolar. That doesn't necessarily mean anything. It's completely unique to each drug about what that actually means as to whether it's loss or gain of function. So by plotting your variant as a ratio against the wild type, it gives you an idea about whether the value is higher than it should be or lower than it should be. And so, for example, if the EC50 value is lower, that means it binds, for example, glutamate or so that it would be glutamate in GLUN2A. It binds it higher, so there'll be a higher affinity, so there'll be too much activity. Or if the ratio is lower, that means that it doesn't bind glutamate well enough, and you'll start getting underactivity. That would be the broadly 50. Okay. Oh, for magnesium. Okay, yes. So so magnesium, as I spoke about, is a very important ion for controlling NMDA receptor responses. And if the IC50 for magnesium goes down, that's going to mean they're under less control, and so they're more likely to be opened by glutamate alone, whereas they're really supposed to be gated by glutamate only when the Ampa receptors have been activated to a certain degree. So yes, all those numbers are important to understand exactly how the receptor's function has been changed.
Thanks, Ian. We have another question. I'll direct this one to Dr. Swanson. So this one was sent to me by a direct message, so you can't see it in the chat. But anyways, it's does gain of function mean that the flow is consistent at a specific rate, or does it fluctuate? And then they had a follow-up question to that. Would gain of function impact minerals within the bloodstream? There's such as fluctuating sodium and magnesium levels that routinely run on the high side, but can spike for no apparent reason. So they wanted to know if those were related.
Yeah, the conductance through the receptor is pretty consistent, I would say. And then a gain of function could be because there's a larger conductance through the receptor, or it could be that the conductance is the same, but it just lasts longer. Those would both be characterized as a gain of function. Or as Ian was just talking about, it could be that the conductance is activated at a lower concentration of glutamate. So you need less glutamate to get the same level of conductance. On the second one, I think it's the plasma levels of sodium and potassium are unlikely to affect the glutamate receptor activation normally because one, they're not in the same, we're talking about a different compartment. So the synapses are in the brain and not exposed to the plasma. And secondly, the way these are activated, as Ian showed, is that glutamate is released from these small vesicles and it goes across a very short gap called a synapse and binds to the receptors, and that's when they get activated. And the concentration that's required to activate those receptors is fairly high in the micromolar range, but it only lasts a very, very small amount of time. So on the order of milliseconds for Ampa and kainate receptors, a little bit longer for NMDA receptors. And so they're designed in their machinery, in the structure, to shut down if they're exposed to constant levels of glutamate. If you have, for example, a pathological condition where, for example, in stroke or ischemia, where the glutamate level rises and stays constant, that actually can be very damaging. So the receptors are not, they're designed to respond to fast pulses of glutamate for the most part, and not so much to constant levels of glutamate.
Thank you, Dr. Swanson. I don't see, oh, whoops, there's another one. Okay, so another person asked, did I understand correctly that there are less drugs available for GRIC variants than for GRIA or GRIN variants? Is that because GRIC2 was only recently detected, or because the kainate receptor is more difficult to treat?
Yeah, that is true. There are fewer drugs available to modify the activity of kainate receptors, and there are a couple points. One is that there are drugs that do that, except that they also inhibit Ampa receptors, so they're not selective between Ampa and kainate receptors. There are drugs for Ampa receptors that will tune up or tune down the activity of those receptors, but for the most part, there aren't equivalent sorts of drugs for kainate receptors that will tune up or tune down in a sort of reversible way, like you would hope for in a drug. And the reason why that is the case is not so much because they were more recently characterized or isolated, or that they're more difficult to treat. It's just that people have not discovered drugs that do that, have that effect on the receptor. There have been many years of trying by drug companies, most recently by Eli Lilly, and they just haven't, for reasons that are not exactly clear, they just haven't had any success in finding those kinds of drugs, even though there's good reason to pursue those compounds as drugs, because kainate receptors are a bit more modulatory in their function relative to Ampa receptors, as Ian pointed out. He left out kainate receptors in many of his cartoons. And that's because they don't perform the same function as Ampa and NMDA receptors at excitatory synapses. And it's been especially for someone in the kainate receptor field, it's been disappointing and a little frustrating that compounds or drugs have not materialized that will do similar sorts of things that drugs for Ampa and NMDA receptors will do.
Thank you. That makes perfect sense. Well, it looks like we don't have any more questions in the chat. Does anybody want to add anything or unmute yourself if you had another question?
I have a quick question. So I wanted to ask Dr. Swanson, so you talked about the limitations of doing the studies on Hek cells. I believe that you have patient-derived iPSCs.
Mm-hmm.
Have you considered making those into organoids and doing the same tests?
Yeah, I think again that's an approach which also is very powerful, but also has its limitations because you can. So just to back up, iPSCs are in stem cells essentially that you can derive from skin cells from a patient, and then they would have the variant that the patient has, and you can induce those cells to form neurons in a dish, different kinds of neurons, and then measure the properties of those neurons and gauge how they differ from an unmodified receptor. And that's a very common way of approaching comparative studies that complements the recombinant recordings and patch clamp analysis that we have. So what is the limitation? The limitation is that those neurons, they spread in a two-dimensional layer. They tend to be homogeneous. So in the brain, we have lots of excitatory neurons connecting with inhibitory neurons, connecting with other sorts of modulatory neurons in very complex, like amazingly complex environment and architecture. That's not reproduced in iPS cell-derived neurons. So there are reasons why it's a useful thing to do, and we have thought about doing that. It's, you know, that we just haven't gotten around to it, I guess, is the answer yet.
Thank you.
Right. Well, it looks like we're out of time and I don't see any more questions in the chat. Thank you so much for your time, Dr. Swanson and Dr. Coombs. You did a fantastic job explaining some really complex stuff in an easy, digestible way for families. And if we receive any more questions by email, I'll direct them to you. And again, we wanted to thank our sponsor, Homology Medicines, and thank you all for your time and attention today.
And thank you, Megan, for putting this together and for your presentation. It's been great. Thank you.
Thanks very much for the invitation. It was great fun. Thank you.